Welcome everyone, and thank you for joining us for the second installment of our workshop series on managing orchards for climate-friendly outcomes. My name is Louise Nelson, and I'm a Professor Emerita in the Department of Biology and co-investigator on this project. As we begin, next slide, let me first acknowledge that UBC Okanagan is situated in the territory of the Salix Okanagan Nation and their peoples. I would also like to acknowledge that you are joining us today from many places near and far, and acknowledge the traditional owners and caretakers of those lands. Next slide. I'd also like to acknowledge that this series is brought to you by the BC Food Web. The BC Food Web is a web portal project that aims to increase access and connection to current research and other resource materials. In addition, it aims to encourage innovation between farmers and researchers for future project collaborations. Today is the second day of our three-part workshop series, which describes the results of a 5-year project funded by Agriculture and Agri-Food Canada through their Agricultural Greenhouse Gases program. Our goal was to investigate how some orchard and vineyard management practices can influence the loss or retention of soil organic matter and nitrogen. Our focus has been on irrigation, but we also considered several other factors, such as mulching. There are two reasons that we focus from the beginning on organic matter and nitrogen. First, they are the two key constituents of healthy and productive soils. Our second reason is that when carbon and nitrogen are lost from soils to the atmosphere, it is through the release of greenhouse gases, carbon dioxide or nitrous oxide. The focus of today's workshop is on the microbial nitrogen cycle and its contributions to nitrous oxide emissions. My colleagues, Dr. Tanya Vogel, Melissa Larrabee, and Catherine Jansen will discuss new methods for quantifying and cycling microorganisms and examine how such factors as soil properties, moisture, carbon and nitrogen amendments, and a nitrification inhibitor can affect the abundance of these organisms and the emission of nitrous oxide in Okanagan soils. Based on this research, we will outline some potential implications for agricultural practices to either mitigate or enhance emissions of the greenhouse gas nitrous oxide. A little bit of housekeeping here. After each panelist has given a brief presentation, we will move on to the Q&A portion of the webinar. If you have questions for the panelists, please navigate to the Q&A tab at the bottom of your Zoom control panel and share them with us. Also feel free to upvote others' questions by clicking on the thumbs up symbol. So I have a few brief introductory slides to introduce the nitrogen cycle to you and some of the concepts that you'll be hearing throughout our presentation. So if I could have the next slide, please. So here is a brief overview of the terrestrial nitrogen cycle. Nitrogen is an essential component for all living things. Unfortunately, with the exception of a few nitrogen-fixing bacteria, the nitrogen that is present in the atmosphere is not available to plants or animals or most other microorganisms. In order to acquire nitrogen and get it into the soil system, there are three different ways which are shown here. It can be through fixation by microorganisms, or in agricultural systems, the addition of fertilizers, either mineral fertilizers or organic matter, which is in the form of plant or animal wastes, compost, mulches, that kind of thing. Once into the soil, the nitrogen is mineralized to ammonium and then cycled through two major processes, which are the focus of our presentations today. Those two processes are nitrification and denitrification. Both these processes release nitrous oxide as a byproduct. As a result, agriculture is a major contributor to nitrous oxide emissions. The organisms that cycle these are important, but difficult to culture and quantify. And so we are looking at different methods for understanding how these organisms are working and their abundance in the soil environment. By studying the environmental factors and agricultural practices that contribute to nitrous oxide emissions, we can determine how best to mitigate the release. Next slide, please. So if you'll bear with me, we're going to look in a little more detail at those two major processes that we're talking about today, nitrification and denitrification. Nitrification is carried out by bacteria and archaea. Archaea are single-celled organisms, very similar to bacteria, but have a different taxonomic basis. And they're able to oxidize ammonium to nitrate through a series of steps, which you can see on the figure here. It's important to emphasize that nitrification occurs under aerobic conditions in the presence of oxygen. So these organisms tend to be active, for example, in well cultivated, aerobic, well-drained soils. The first step, the conversion of ammonium to hydroxylamine, is catalyzed by the enzyme ammonia monooxygenase, and it's coded for by the gene AMO-A, and you will hear that from several of our speakers today. Bacterial AMO-A is, we will call B-AMO-A, and archaeal AMO-A, which is present in the archaea, we'll call A-AMO-A. As you can see from the slide, nitrous oxide is produced as a side reaction from hydroxylamine. We can use the abundance of AMO-A, we can measure that using techniques that Dr. Vogel will talk about. We can measure its abundance in soil, and this gives us an indication of the potential for nitrous oxide emission by the nitrification process. Next slide, please. The other side, and the next process that occurs, nitrate that has formed can be recycled back into the atmosphere by the process of denitrification. This occurs under low or no oxygen conditions, and it occurs with a series of steps, gradually, with nitrous oxide being the penultimate product, and then nitrogen gas. Next slide, please. We can look at a number of genes along this pathway. One of these, the nitrate reductase enzyme, codes for the reduction of nitrite to nitric oxide. This is coded for by two types of genes in soil organisms, near S and near K. And again, by measuring the abundance of these two genes, it gives us an indication of the potential of denitrifiers to produce nitrous oxide. Next slide, please. The final enzyme in the pathway, nitrous oxide reductase, which reduces nitrous oxide to nitrogen gas, has two forms among soil microbial populations. The genes are called NOS-SED1 and NOS-SED2. And we can look at the abundance of these genes as an indication of the potential to reduce nitrous oxide, which is a potent greenhouse gas, to the inert gas N2. Thank you. Next slide, please. So our approach in this study has been to look at what are the factors that are influencing nitrous oxide emissions from Okanagan orchard and vineyard soils, and are there practices that can minimize these emissions. The first step that we looked at was finding ways to quantify soil nitrogen cycling populations. And we used a new method, which is DNA based, so that we don't need to culture or isolate the organisms, but simply to extract their DNA from soil and look at what genes are present. And Dr. Vogel will be discussing those. And then we looked at factors that drive nitrous oxide emissions in these soils. To do this, we used microcosms, i.e. we brought soil indoors, placed it under controlled conditions, and we varied key factors one at a time and looked at what the effect was on the emissions of nitrous oxide or as well on the soil end cycling populations. So with that, I'm ready to move to Dr. Vogel's presentation. And I'll introduce her first. Dr. Vogel is a research associate in the Department of Biology, studying microbiology, plant pathology, and the soil nitrogen cycle. She developed DNA-based methodology to quantify nitrifier and denitrifier abundances in soil. and she's currently studying grapevine crown gall, but will talk to us about her nitrifying project right now. Thank you.
Tanya is actually having internet problems, so we are just going to move on to my part, Louise.
Oh, okay, sorry. Well, I will now introduce you, Melissa. So I'm very pleased to introduce Melissa Larrabee. She's a research technician in the Department of Biology with an MSc in biology. She is studying how different soil applications affect greenhouse gas emissions from agricultural soils.
All right, thank you, Louise. So I'm going to do my best to explain this without Tanya's introductory presentation. So I'm going to be talking about the effect of soil treatment on nitrous oxide emissions from 3 soil types in the Okanagan. So this should be familiar to you by now, so I'm going to try to keep it fairly brief. We know that nitrous oxide is a potent greenhouse gas produced in agricultural soils through the processes of nitrification and denitrification. Like Louise went over, nitrification is the conversion of ammonia into nitrate, and it produces nitrous oxide as a byproduct. It uses bacterial AMOA and archaeal AMOA, and it occurs in the presence of oxygen. And if you think about it, it makes sense because you're taking ammonia, which has no oxygen atoms, and you're adding 3 oxygen atoms onto it to create nitrate. So you need oxygen. Denitrification, on the other hand, is the conversion of nitrate into 3 gases: nitric oxide, nitrous oxide, and dinitrogen gas. It uses enzymes and genes such as NIR-S, NIR-K, NOSED1, and NOSED2. And unlike nitrification, denitrification is occurring mainly in the absence of oxygen. And the other thing I want to draw your attention to is that the end product of nitrification, which is nitrate, is the starting substrate for denitrification. So in this way, nitrification feeds denitrification. So we had 3 goals of our study, and that was to assess the effect of moisture content, nitrogen content, and carbon content on nitrous oxide emissions and nitrogen cycling gene abundances for nitrification and denitrification in 3 different soil types. So we hypothesized that as we increased moisture content, we were going to see an increase in nitrous oxide emissions. And that's because, like us, bacteria need water to grow. So as you add more water, they're going to be able to grow and double. And also that as you're adding more water to the soil, the spaces in the soil that would normally have air are now being replaced with water. So you get these pockets of oxygen and no oxygen, allowing nitrification and denitrification to occur at the same time. So we also expect an increase in nitrifying and denitrifying gene abundances. To look at the effect of nitrogen content, we added different levels of urea. Urea breaks down into 2 molecules of ammonia, and if you remember, that is the starting substrate for nitrification. So by adding urea, we are feeding nitrification, and we are going to see an increase in nitrous oxide emissions, as well as an increase in nitrifying gene abundances. We can expect either an increase in denitrifying gene abundances because nitrification feeds denitrification, or even a decrease if there isn't enough carbon to stimulate the denitrifiers. Which leads me into our 3rd experiment, which was looking at carbon content. We added different levels of compost. We used Glengrow, so that is a green compost made of yard waste. And based on the research, compost has been shown to either increase or decrease nitrous oxide emissions. So we came into this kind of expecting almost any result with that. Before I get into our experimental design, I want to talk about the soil types we studied. For those of you who were at the soil carbon session yesterday, we sampled 3 of the same soils that they sampled, and the soils we sampled all came from apple drip locations. So we had the first that we call AAD from Armstrong, GAD from Glenmore, and SAD from Summerland, or as the soil carbon group calls Penticton. They're all within the same class of loam, but they do vary a little bit. There's no difference in silt between these types. AAD has more sand than GAD, and GAD has more clay than AAD or SAD. And as for the water-filled pore space, which is the moisture content of the soil, we determined that at the time of sampling. And as you can imagine, it is influenced by recent irrigation and rainfall. And for the urea and compost experiments, we changed the soil moisture to match that we measured in the field at the time of sampling. So how did we do all this? Well, we first had to go to all these orchards and collect the soil. We brought it back to the lab where we dried it and sieved it, and we added it into flasks to create what we call a soil microcosm. So this is a very controlled, simulated soil environment. And then we did our 3 experiments. So our moisture experiment lasted 9 days, our urea experiment lasted 9 days, and our compost experiment went on for 30 days. We measured our nitrous oxide emissions throughout the experiments, and we measured gene abundances and soil properties at the start and end. For gene abundances, we have the AAD soil type completed. We have most of GAD completed, but there's still another experiment, and SAD is still in progress. So I'm just going to present to you the data we have. Before I get into the data we have, I have to explain 2 terms called total abundance and relative abundance, and I'm going to try to use a metaphor that's hopefully relatable to you. So imagine you have 2 apple blocks and they need to be harvested. You invite your friends to come help you, and they split into 2 groups. Group 1 was able to pick 100 pounds of apples, and group 2 was able to pick 50. And if you only care about total abundance of apples, you would think that group 1 wins because they picked 100 pounds, that's more than 50. But if you cared about how much each of your friends actually contributed to your harvest, you would need to know how many friends was in each group. So if 10 of your friends picked 100 pounds, that means about each person picked 10 pounds each. And if 5 of your friends picked 50 pounds, that also means each of your friends picked about 10 pounds each. So we have a difference in total abundance, but not relative contribution. So the only reason group 1 picked more is just because there was more of them, not because they were working harder. This is in contrast to if your 5 friends had picked 100 pounds. Now the total abundance is the same between 2 groups, but your 5 friends contributed more apples. I hope that makes sense as we keep going. This is looking at the effect of moisture content on nitrous oxide emissions. So on the y-axis here, we have accumulated nitrous oxide through the duration of the experiment. We have our soil type on the x-axis, and we have our treatments here in the legend. So 0% is our control, 30%, 50%, and 80% water-filled pore space. And all of our soil types showed a similar response where we have 80% producing significantly more nitrous oxide than 50 or 30, which are producing the same, but are producing more than our control. And for reference, treatments within a soil type that have the same letter here are not significantly different. Looking at the effect of moisture on total nitrifying gene abundances, so again, that's the total, we have total bacteria, total archaea, bacterial AMOA, and archaeal AMOA. And we can see for each of them in each soil type, we're getting a significant increase in total gene abundance as we increase our moisture content. And the exact same thing is seen when we look at our denitrifying total gene abundances. So here we have near S, near K, NOS-SED1, and NOS-SED2. And again, we're getting this stepwise increase where total gene abundance is increasing as we increase moisture content. And this is what we expected, because as I said, as we're increasing moisture content, we're stimulating these bacteria so they're growing in abundance. And this is the equivalent of picking more apples. But when we look at the relative gene abundance, so asking about contribution, for the bacterial AMOA and the archaeal AMOA, these are the nitrifying genes, we actually get no difference between treatments in relative gene abundance. And we see the same thing when we look at denitrifying gene abundances. So again, this relative gene abundance is not being changed by moisture treatment. So what we know is that as we increase moisture content, we increase nitrous oxide emissions, and we increase total microbial abundance, because as we increase moisture, we're just increasing more bacteria that are there. And that is why we're getting an increase in nitrous oxide emissions. Moving on to our next experiment, which was looking at the effect of nitrogen content on nitrous oxide emissions. This graph is set up in the exact same way, except now our treatments are urea and it's 0, so that's our control, 25, 50, or 75 milligrams of urea added to 50 grams of our microcosms. And here we actually see a different pattern between soil types. So for AAD, we see a stepwise increase in nitrous oxide emissions as we increase the urea content, whereas for GAD, regardless of the urea content, it's emitting about the same amount of nitrous oxide. And we can see that for SAD as well, where the two highest levels of urea are emitting the same level of nitrous oxide. So here we're looking at the effect of nitrogen content on nitrifying gene abundances. There is no effect on total gene abundances, so I'm not going to show that data. So this data here is relative gene abundances. And on the left, we have bacterial ammo A, and we can see that for both AAD and GAD, the total gene or the relative gene abundance is increased in our urea treatments as compared to our control. And then in the case of our kale ammo A for GAD, we actually see that its relative abundance significantly decreased at the highest level of urea compared to the control and the lowest level. And this actually makes sense because archaeal nitrifiers are very sensitive to ammonia and it's toxic to them. So this is not a surprising result. Looking at the denitrifying gene abundances, again, this is only looking at relative gene abundances because there is no effect of total. For near K in AAD, we can see that it was significantly increased compared to the control and the lowest level. And for NOSTED2 and GAD, we see a significant decrease in the 25 milligram treatment compared to the control. I'm not exactly sure what's going on here right now, so I don't have an explanation for you at this time. In summary, by increasing nitrogen content, we saw an increase in nitrous oxide emissions, and we saw an increase in relative nitrifier abundance and denitrifier abundance, which is likely causing this increase that we saw in nitrous oxide emissions. On to our last experiment, looking at the effect of carbon content on nitrous oxide emissions. The graph again is set up in the same way, but here are our treatments, 0 grams, which is the control, 0.5, 1 or 1.5 grams of compost, again added to our 50 grams of soil in each microcosm. And there is no effective compost treatment on any of the soil types for nitrous oxide emissions. But we can see that in general, GAD is emitting more nitrous oxide than AAD or SAD. This hasn't been statistically assessed though. Looking at nitrifying gene abundances, on the left we have total and on the right we have relative, and it goes bacterial ammo A and then archaeal ammo A. Either way, there was no effective carbon content or compost treatment on any of these gene abundances, whether it's total or relative. And the same can be said for the denitrifying gene abundances. So again, we didn't see any effect of compost treatment on total or relative gene abundances. So in the case of adding compost, we didn't see an effect of nitrous oxide emissions and we didn't see an effect on gene abundances. We've only looked at AAD though. We're still collecting data for GAD and SAD. That may be different. So to kind of bring it all back together, in the case of our moisture content experiment, we saw that wetter soils increase nitrous oxide emissions for all the soil types we studied. So it's really important for us to pay attention to our irrigation and our soil saturation. For our urea experiment, we saw that adding urea significantly increased nitrous oxide emissions, but perhaps the most interesting result is that the total nitrous oxide emitted was similar between their urea treatments for GAD and SAD. So perhaps in these soils, just the act of adding urea is more important than the amount that is added. And in the case of compost, we didn't see any effect. And perhaps this could be because the study was too short. Maybe if it went on longer, we would see a greater effect. Or the fact that this study did not compare its results to a fertilized treatment. And perhaps that's why we're not seeing any increase or decrease in compost. And Catherine will be able to talk a little bit more about that in her microcosm. And so with that, I thank you for bearing with me and getting through all that data. I know it was a lot. And thanks to the Nelson Lab, AGGP2, all of the orchards that allowed us to sample their soil, and Craig Nickel for helping us run the gas chromatograph.
Okay, thank you very much, Melissa. And now we'll go back to Dr. Tanya Vogel. I think she's rectified her problems with the webinar. So as I mentioned, Tanya is a research associate in the Department of Biology, studying microbiology, plant pathology, and the soil nitrogen cycle. And she's going to tell us how we have developed methods to quantify those nitrifying and denitrifying populations. Thanks, Tanya.
Okay, thank you, Louise. And sorry everyone for earlier. I had some internet problems, but it seems to be working now. Next slide. So before I get into the science, I would like to get you started with a little trivia. Did you know that there are more living individual organisms in a tablespoon of soil than there are people on Earth? So next time you're outside, just grab a handful of soil and try to imagine the billions of microorganisms that you're holding in your hands. And you know that agriculture is the only essential industry on Earth. And that is because soil provides us with feed, fiber, food, and fuel. And did you know that almost all of the antibiotics that we take to help us fight infections were actually obtained from soil microorganisms? So not only does soil provide feed, fiber, food, and fuel to us, but it also keeps us healthy. And did you know that soil is a non-renewable natural resource? So soil is a finite resource, and that means that its loss and degradation is not recoverable within a human lifespan. It's a very highly valuable natural resource, but it's often overlooked. Next. However, soils and agriculture are also a major contributor for the emission of greenhouse gases, such as carbon dioxide, water vapor, methane, and nitrous oxide. Nitrous oxide is especially important as 1 unit of nitrous oxide has a warming potential that is about 300 times higher than that of 1 unit of carbon dioxide. And most of the nitrous oxide emitted by humans, is shown here in this figure for the United States, is generated by nitrogen fertilization of soils. On the flip side, the same agricultural soils also have the potential to reduce, or like we say, mitigate greenhouse gas emissions through different management strategies, such as reducing the amount of the fertilizer that's being used, crop rotation using legumes, no tillage, or switching to organic amendments instead of using synthetic fertilizer. Organic amendments would release nitrogen slower and prevent excess nitrogen and nitrogen leaching. Next. So for example, as part of the Agricultural Greenhouse Gas Project 1, we have shown that the addition of mulch has reduced nitrous oxide emissions in vineyards and in apple orchards here in the Okanagan. Next. So the objectives for the current project were what soil treatments can be used to reduce nitrous oxide emissions from soil. And specifically, we set out to study which treatments are suitable on a regional scale here in the Okanagan, as the effects will likely be specific to the environmental conditions and the local soils that we have. So I'm not going to talk about this objective here, as Melissa has already presented some and Catherine will add to this in a few minutes. And secondly, how can we measure what effects any of the potential soil treatments can have on the processes that generate nitrous oxide? So Louise has told you earlier that most nitrous oxide is generated by bacterial and archaeal microorganisms, and mainly by 2 processes, nitrification and denitrification. So this means that for our research, we needed a methodology to quantify the abundances of these microorganisms in the soils in response to any soil treatments that we may apply. Next. So this slide shows some of the billions of microorganisms I've been talking about before. They all contain DNA, and that's shown here in yellow. That DNA stores genetic information that is necessary to build and maintain an organism. Genes that are indicated here in blue are areas on that DNA that contain the information for specific products, for example, for genes that are involved in nitrification or denitrification. And in the lab, we can collect the soil and extract the DNA. And we only needed a methodology to quantify the gene numbers in that soil. Or a better word would be the abundances of the genes that we're interested in. Knowing the gene abundances in the soil allows for prediction of the processes that occur in the soil in response to any of the soil treatments. Next. The instrument we chose to use for our experiments is called droplet digital PCR. And that's a relatively new technology that can quantify specific genes. So I'm not going to go into the technical details on how ddPCR works, but just keep in mind that we are using ddPCR here to quantify the genes that are involved in nitrification and denitrification. This method has some advantages over some other older technologies, especially when DNA samples are used that were extracted from soil. ddPCR is a lot more sensitive, and any of the inhibitory substances that are present in the soil have less of an effect for quantification here. Next. So we did develop the assays for 8 specific genes, and those genes are shown here in the table, and all are important and involved in the nitrogen cycle. 16S bacteria and 16S archaea measure the abundance of all bacteria or archaea in the soil. Near K, near S, B, MOA, and A, MOA measure the potential to produce nitrous oxide, and NOZ, clade 1 and 2 measure the potential to reduce nitrous oxide. The individual assays for each gene needed to be developed and optimized using the ddPCR before we can apply and use it in actual research. And because no other researchers has done this previously for the genes shown here, we developed the assays so that the assays can then be used by other researchers and specifically by Melissa and Catherine in their studies. Next. So I'm not going to talk about all the technical details and what's involved in developing ddPCR assays, but if you are interested in learning more about the development, then I invite you to read our paper that was published last year in the Canadian Journal of Microbiology. Next. So now we had the method developed, but we needed to confirm that the method actually works when real soil samples were being used. And if the results that we see when we use this ddPCR with the soil samples follow any known pattern that were described by others. So in short, we needed to validate our method. To do this, we collected soil samples from 2 vineyards in the Okanagan Valley. And the table here shows the differences between the 2 vineyards, and they were mostly between the location and the age. So I want to emphasize here that we actually did not apply any treatments to those sites. Those were regular vineyards with untreated soil. Next. We went 5 times into each of the vineyards, and we sampled 6 vines. And we took 2 30-centimeter cores at each vine. And we then took the soil into the lab and separated the roots where the rhizosphere soil is attached. And we only needed 0.25 grams of this soil to extract DNA. So this is such a small amount. We then ran ddPCR on all of those 8 genes, but we also analyzed the soil for carbon-nitrogen ratio, soluble organic carbon, ammonium, nitrate, pH, soil temperature, and soil moisture. And we also looked at air temperature and rain events. And after collecting all the data, we were using a statistical analysis program called R. Next. So this slide shows the different growth stages that we sampled at. The first date was in May 2nd at bud break. The second time we went out in June 20th when at bloom. In July 6th, berries were pea-sized, and in August 28th, veraison happened. And then again, we sampled the last time in October at harvest. Next. So this graph visualizes the gene abundance data for all the genes from Vineyard 1. Each group of 5 bars represents one specific gene, and each color represents one growth stage. For example, the green bars show the gene abundance at bud break. The arrows that are next to the gene numbers below indicate if the gene is involved in the production or in the reduction of nitrous oxide. You can see that the highest numbers were for total bacteria, followed by the total archaeal group. But overall, there's very little differences between each of the growth stages. Next. These are the results for Vineyard 2, and they are very similar results to Vineyard 1. So again, the total bacteria were the highest, followed by the 16S archaea. To better assess the gene abundance data, we did use a statistical model, and we would that not only takes the abundance data as shown here into account, but also some other factors which can influence the data. So this includes the soil properties and the environmental data, such as temperature and precipitation. Next. So these are the results when we take both the gene abundances and the soil properties into account. Only one gene, Nitrous, that takes part in denitrification, is affected by the location of the vineyard. And gene abundances also change with time. And this is expected as the microorganisms continue to grow unless there is a shortage of nutrients, for example. For example, 16S bacteria are more abundant at the pea-size berry and harvest stage compared to bud break. And then also some of the denitrification genes were higher at all growth stages compared to bud break. Next. Soil properties also affected gene abundances, and all effects that we've seen are negative. And that means that the gene abundance decreases if the value for the soil property increases. For example, all gene abundances decreased with an increasing pH. So when we compared our results to others, we found similar results. For example, the archaeal abundances 16S and AMOA are negatively related to the carbon-nitrogen ratio and ammonium content. So the comparison of our data to known results showed that our ddPCR method is a valid methodology able to detect any changes in those gene abundances. Next. So as a summary, we developed a ddPCR method here to quantify genes that are involved in nitrification and denitrification, and we validated the methods using local soil samples. We found that we can assign genes into 3 groups where the abundance is affected by soil properties and growth stage, by soil properties only, and by soil properties and the location of the vineyard. So now we have a tool in hand that we can use to study the effects of any soil treatments on the processes involved in generating or in reducing nitrous oxide. And with that, I would like to turn it over. No, next slide, please. Got my thank you slide. Yeah, with that, I would like to thank you and the Agricultural Greenhouse Gas Program and also Quailsgate, who allowed us to take samples from their vineyard.
Great, thank you very much, Tanya. And now we will move on to the next speaker, Catherine Jansen. Catherine is a graduate student at the University of British Columbia working on her master's. Her research focuses on the use of different soil additives and practices to reduce the production of the greenhouse gas nitrous oxide from agriculture. Take it away, Catherine.
Thank you. Hello, everyone. As Dr. Nelson said, my name is Catherine, and today I'll be talking to you about the impact of soil management on emissions of the greenhouse gas nitrous oxide. So for a quick overview, I'll just give a brief introduction first about nitrification inhibitors. This will be followed by my methodology and the results of my experiment, before finally finishing with my conclusion as to what these results mean. And to introduce each section, I'm just going to be reading a brief quote about soil. So first off, to feed our people, we must first feed our soil. Olusegun Obasanjo. So here in Canada, BC is the largest fruit growing province, with the Thompson Okanagan specifically having the second most abundant amount of cropland dedicated to fruit and nut growing at about 40%. Most of this is made up by apples, sweet cherries, and grapes, though there are many other crops such as many stone fruits like peaches, apricots, and plums, and there are also various different nuts that are grown. And so with any kind of agriculture comes the need for different soil additives to ensure optimal nutrient availability for your crops, which helps in production efficiency for those crops. This includes the use of fertilizers and composts. And these additives can cause an increase in the emission of greenhouse gases, depending on the types that you use. Because of this, there are additional additives, such as nitrification inhibitors, that may be added to mitigate the effect of these fertilizers and composts to create those greenhouse gases. And so specifically, nitrification inhibitors work to reduce the production of nitrous oxide and/or nitrate by stopping the process of nitrification from going to completion. This can alter microbial communities and soils from altered energy acquisition, and the effectiveness and toxicity of these nitrification inhibitors varies due to many factors, one being just the composition of the chemical itself, and then also the longevity in the soil. 3,4-dimethylpyrazole phosphate, which is a mouthful, so I've written it as DMPP for short here, is a nitrification inhibitor that specifically affects the bacterial form of the enzyme ammonia monooxygenase to inhibit the first step of bacterial nitrification. And so as you can see here on this slide, ideally this prevents the production of nitrifier nitrous oxide and reduces the amount of nitrate produced as well, which would otherwise feed into denitrification and potentially contribute to denitrifier nitrous oxide. I do want to stress as well that although it inhibits bacterial nitrifiers, archaeal nitrifiers are another story because they have a more sensitive form of the enzyme. But ultimately, overall, I wanted to know if these methods were viable for upscaling. How would various treatments involving nitrogen sources that feed into either nitrification or denitrification or both be impacted by the addition of this nitrification inhibitor, DMPP? Specifically, I wanted to know how nitrous oxide levels would change, and I wanted to know how it would affect the different microbial nitrogen cycling gene abundances. I also wanted to look at how nitrous oxide emissions would change with added compost. And so my hypotheses were that when adding nitrate, this would stimulate the denitrifiers because it's providing a substrate for their energy processes and increases nitrous oxide emissions. I also expected the addition of ammonium to stimulate denitrifiers. However, this would be an indirect effect from actually stimulating nitrifiers because that is the beginning substrate for nitrification. And so it's kind of like if you went out to buy food for yourself, but then your friend ate the leftovers. It wasn't really meant for them, but because you had it, they benefited. And ultimately, this would lead to an increase in nitrous oxide as well. When DMPP was added to ammonium, I expected bacterial nitrifiers to decrease, causing a decrease in nitrous oxide and nitrate produced by nitrification, which could also decrease denitrifier nitrous oxide. And so when added to nitrate, the DMPP added to nitrate, I didn't expect to see the denitrifiers be affected much, if at all, but that there may be slightly less emissions if it prevented the use of pre-existing ammonium in the soil by nitrifiers. However, I thought this would be insignificant if it was happening. The soil is the great connector of our lives, the source and destination of all. Wendell Berry. And so for this experiment, the soil used was collected from bare cherry plots at the Summerland Research and Development Center, which were in the past treated with calcium nitrate fertigation and microsprinkler irrigation. However, this past year, a different ammonium-based fertilizer was also applied, due to reduced occupancy at the center. So my experiments used the soil to create microcosms, each with about 50 grams. And my trials included varying amounts of ammonium, nitrate, and DMPP at 50% the recommended dose for the first trial. And then the second trial used ammonium, compost, and DMPP at 100% the recommended dose. And so after the microcosms were set up and after sealing them, they were sampled incrementally using a gas needle, and these gas samples were read using a gas chromatograph. And on the first and last day of these microcosm incubations, soil was sampled and used for analyses. This includes DNA extraction, which was then used to quantify nitrification genes using the ddPCR method that was explained earlier by Tanya. Soil pH and different carbon and nitrogen values were also assessed. Combustion was used to identify carbon-nitrogen ratio, and colorimetric methods were used to quantify nitrate and ammonium, such as the ones displayed on the right of the screen. The nation that destroys its soil destroys itself. Franklin D. Roosevelt. And so for my results, this plot shows the total emissions for the first microcosm trial conducted. You can see on the next slide that the treatments with nitrate added had significant increases in nitrous oxide emissions compared to the bare treatment. However, the treatment with ammonium alone did not have any significant changes from bare, meaning that the change in the treatment with both ammonium and nitrate was likely more contributed to by the nitrate than the ammonium. When DMPP was added to the treatment with nitrate, there was no significant reduction in nitrous oxide emissions from the treatment with the nitrate alone, which was expected. Interestingly, there was also no effect with the addition of DMPP to the ammonium treatment, which was unexpected, but this may be due to the fact that the microbial community was often not exposed to ammonium because it was usually fertigated with calcium nitrate, meaning that the microbial community may have been characterized by groups with increased sensitivity to ammonium, like archaea. And so finally, as noted earlier, when both fertilizers were mixed, there was a significant increase in nitrous oxide emissions compared to the bare treatment. However, when the 50% recommended DMPP was added to this, the increase in emissions was significantly higher than all of the treatments. For my second trial, nitrous oxide emissions increased significantly when fertilizer was applied with compost compared to the bare and fertilizer alone. So you can see that adding just the ammonium did not change the emissions significantly, but when compost was added in conjunction with the fertilizer, it did increase emissions. And so this does make sense as well, because if the compost had a high amount of readily available carbon, but not much nitrogen, the increased nitrogen from the fertilizer can increase the activity of microbial groups to use that nitrogen instead of sequestering it. Additionally, it would have increased organic carbon for denitrifiers who use that in denitrification. When DMPP was added to this treatment at 100% the recommended dose, the amount of nitrous oxide was significantly decreased back to levels that resembled bare soil. When you compare the first and second trial, the similar treatments of the two trials, it would seem that the addition of DMPP does cause a more significant reduction in emissions when added at a rate of 100% the recommended amount, as in trial 2, but it's not significantly different from the ammonium applied alone within the same treatment. So it may not be worth adding it if you are applying just the monoammonium by itself. The results for the gene abundances in the first trial showed that the relative amount of bacterial MOA was not significantly different in any comparisons of interest. So that means within any treatments within the same day or within any days in the same treatment. However, the relative amount of archaeal MOA did show an interesting pattern between the genes and the gas emissions. So the 3 treatments here that included nitrate all had significantly less relative archaeal MOA compared to the bare treatment. The significance pattern was the same for the reduction in relative archaeal MOA and increases in nitrous oxide emissions compared to bare of those treatments. So both fertilizers mixed had a more significant impact than nitrate alone on the gene abundances and gas, which had a more significant impact than the nitrate mixed with the DMPP. However, one of the treatments did not follow this pattern, which was the treatment with all 3 additives, showing a significant increase in nitrous oxide emissions compared to all other treatments, but no change in the relative archaeal MOA abundance or bacterial MOA abundance, meaning that it may be the denitrifiers that are contributing to this result, and more analyses are required for adequate interpretation. This includes quantifying the denitrification genes near S, near K, and NOSED 1 and 2 to identify any relationships between them and the nitrous oxide emissions measured. And so moving into the conclusion, out of the long list of nature's gifts to man, none is perhaps so utterly essential to human life as soil. Hugh Hammond Bennett. So in conclusion, this research found that nitrous oxide emissions may be regulated by the relative abundance of archaeal MOA, which may be decreased by nitrate additions. Previous studies have found reduced emissions when archaeal nitrifiers make up the majority of nitrifier populations. Additionally, DMPP may not be optimal for summerland soils to reduce nitrous oxide with ammonium fertilizer alone. And this may be due to the unique characteristics of each soil and its history, including but not limited to the types of fertilizer used, the fertilizer application method, irrigation technique, mulch applied, etc. Ultimately, every soil is unique, and so more tests need to be done to characterize best management strategies, not only for each soil type, but for different soil histories. And with that, I would like to take a moment to thank everyone who's helped me with this project, including Melissa, who's helped me every step of the way, my supervisor, Dr. Nelson, and our entire lab at UBC Okanagan in the Braze department. I'd also like to thank the Summerland Research and Development Center for allowing us access to the field, and Kirsten for her help with our tasks there. Finally, I'd like to thank the facilitators at Agriculture and Agri-Food Canada who've supported us with funding. And with that, I will pass on the rest of the acknowledgements to Dr. Nelson.
Okay, thank you very much, Catherine. Catherine's and Melissa have accumulated a huge amount of data, and I think they've done an excellent job in trying to winnow it down to make it more easily understood by everyone. And just to go over again, we're very grateful to the Greenhouse Gases Program of Agriculture and Agri-Food Canada for supporting this project. The Summerland Research and Development Center, who have been instrumental in helping us with sampling, with some of the gas sampling, which we weren't actually able to talk about today. Also the orchardists, in particular, the Kidston Farm and the Dornberg orchards, who allowed us to go in and sample soil, Quails Gate Winery for Tanya's validation studies, and also a number of people at UBC Okanagan. Dr. Jones, who is the chief investigator on this project, Andy Midwood, also helping with some of the soil sampling, Craig Nickel with the GC gas analyses, and a number of other students who helped. These are quite labor intensive, some of these projects, and so we're very grateful to them. And then lastly, to the BC Food Web, Jessica and Merid, who have helped facilitate this presentation today. I think we are now ready to move to the question answer, but a very nice quote by Catherine here, which I will read to you. Good farmers who take seriously their duties as stewards of creation and of their land's inheritors contribute to the welfare of society in more ways than society usually acknowledges or even knows. These farmers produce valuable goods, of course, but they also conserve soil, they conserve water, they conserve wildlife, they conserve open space, they conserve scenery, by Wendell Berry. So again, a thank you to all of the farmers and growers, orchards and vineyards who are involved in this important process, and we really appreciate your help in this study. Okay, I think we're ready now to look at some questions. So the first question, was there any consideration of the impact or presence of mycorrhizae? I will say that we haven't directly looked at that. Mycorrhizae are not nitrifiers or denitrifiers, although they help to decompose more complex nitrogen sources in the soil, and they also facilitate the uptake of ammonium and nitrate by plants. I don't know whether anyone else in the group here would like to add anything to that. No?
I do have something that I read before. While it's not specific to the ddPCR that was being talked about while the question was asked, there have been studies that have found that the abundance of different nitrifying and denitrifying populations change the closer they get to the mycorrhizae, becoming more and less abundant in certain situations depending on soil saturation and different nitrogen and carbon additives to the soil, as well as the crop type. But other than that, I haven't looked into the specific effect of mycorrhizae.
Okay, thank you. The next question, is there a possibility in the future for gene treatments to decrease nitrification and denitrification genes? Well, I think we heard from Catherine about the use of nitrification inhibitors, and those are commercially available, but as she indicated, there are certainly changes there in terms of their efficacy in different soil types and under different environmental conditions. In terms of gene treatments, I'm not sure if we can say that for sure. These are important processes. We need these organisms to cycle the nitrogen, but perhaps we can modify their activity by different treatments.
Yes, one such instance where there have been a couple trials so far has been using microorganisms specifically that carry the gene NOSED2. So there's two types. The first type is found in normal denitrifiers and is often actually lacking from them, which means that these are denitrifiers that can't complete denitrification. Whereas the second version of the nitrous oxide, the NOSED, actually is found alone in a lot of microorganisms, meaning they can't produce nitrous oxide, but they can break it down. And so a few studies have found when they added microorganisms that had this gene specifically to soils, they did see reductions in the amount of nitrous oxide produced and an increase in their relative contribution to the community as well.
Yeah, those are good points. Another approach would be, once you've identified those groups of organisms, would be to add substrates that they can use specifically, and so that would enhance their presence in the soil environment. Anybody else wanted to comment?
Yeah, so a gene treatment would mean that the plant or the microorganisms would have to be genetically modified, and then this will raise questions about acceptance with growers and consumers. But we can definitely look into some enrichment like what Catherine said. So using some microbes that carry those Z genes that are important and try to enrich those into soils.
Great. Thank you. Okay, question number 3. If we are looking to decrease emissions, is there a way to implement solutions in a cost-effective, relatively simple way on farms? I'm going to ask maybe Melissa and Catherine to comment on that question.
Would you like to go first, Melissa, or do you want me to?
Maybe you take it away. You've been working with the nitrification inhibitors, so.
Yeah. I would have to say it depends on the soil type. That's kind of been the theme of what we've been studying. Every soil is very unique. A soil, a kilometer away could be completely different from where it is, where you're standing at a given moment in time. And they're super heterogeneous as well. So you really need to get down to the specifics of the soil type to really even try to come up with a method to help reduce emissions. And nitrification inhibitors don't seem to be optimal for summerland soils, but I think it would be valuable to try and test this specific inhibitor in other soil types, maybe ones that have higher amounts of clay or ones with a soil history that includes more application of ammonium. So the longer a certain fertilizer treatment has been applied, the better adapted those microbial communities become at using it. They've actually tested urban soils versus agricultural soils using specific fertilizers, and they found that agricultural microbial communities were able to more quickly use that nitrogen source than urban soils that hadn't been exposed to it before and produced more nitrous oxide. And so when you identify which fertilizer you're using, you can potentially sort of garner that treatment more specifically to that one area. So it's not just about soil type, it's also the soil history of that soil as well.
Thank you. Melissa, did you want to add something?
I mean, it really comes down to make sure you're applying the right fertilizer and at the right rate. And like Catherine said, it involves testing your soil and knowing what you already have before you apply something else.
Right.
Also,
Yeah.
low quality composts seem to be good at reducing nitrous oxide emissions, but in order to use those effectively and help keep nutrients in the soil, you would have to already have soil that is relatively well off nutrient wise in some instances. So it would kind of be a give and take.
Right. And I think, as Melissa had mentioned too, the composts tend to release the nitrogen more slowly, so that may also be helpful in overall reducing the nitrous oxide emissions. Okay, I think we'll move on to the next question. Are there some microorganisms that produce less nitrous oxide but still offer benefits to the plant? Could we change the species composition of our microorganism network? Oh, that's a really good question.
Yeah,
that
was kind of what we were talking about.
Sorry, go ahead, Catherine.
No, sorry. It was kind of what we were talking about, increasing that proportion of those microorganisms that do harbor that second type of the NOSED, because that will help reduce emissions and you will still have the diversity there rather than just trying to get rid of the ones that are causing the nitrous oxide emissions. So if you can create a way to actually facilitate those microorganisms to stay there is the question. A lot of the times when you inoculate soils with different AMF or microbial inoculations, they have a hard time establishing if the conditions aren't ideal because other microorganisms are already adapted to that soil, so they're able to outcompete them. So there's a various different methods that you can use to help and try and keep them there, but it would probably require repeated inoculations over time.
Right.
Yeah, there's a related area of research with plant growth promoting rhizobacteria, which are often inoculated to enhance like nitrogen uptake efficiency and things like that. One study I read actually bacterial impregnated fertilizer, which means the bacteria and fertilizer were mixed together. And it actually significantly increased nitrogen uptake by the plant, which in turn may be helping prevent nitrous oxide emissions by getting that nitrogen into the plant before other microbes have the ability to use it.
Right, and those organisms are of particular interest because they colonize the root surface, so they can direct the nitrogen more easily and efficiently to the plant roots. Okay, the next question. Why is this of specific interest for orchards and vineyards? I think I would answer by saying that most of the work on nitrous oxide emissions, greenhouse gas emissions in agriculture have been done with annual crops. And really, this is a very different system when we have perennial orchards or vineyards. So these soils aren't tilled as often, they're not treated in the same way. There's not as much disturbance in the soil microbial populations as you would get in annual systems. And so there's actually been very little work done so far around the world on these perennial fruit crops or grape vineyards. So I think what we're doing is important to add that information to the world database to help more accurately determine the potential for nitrous oxide emissions on an annual basis, and to understand how we can better approach the issues here. I think in general, and others can correct me if I'm wrong, but that orchards and vineyard systems tend to emit less nitrous oxide, but it depends a lot on how much fertilizer is being added to these systems. And that's a major problem. Some of the reports I've seen out of China, where they're also doing some work in orchards, they're using much higher levels of nitrogen fertilizer than we are here. And so as a result, they have an even greater problem with emission of nitrous oxide. Does anyone else want to add to that?
Potentially tilling the soil, like in annual crops, can also help reduce nitrous oxide emissions. It's not good for soil degradation, but it can help reduce those emissions. The trade-off is generally when you have low nitrous oxide emissions, you tend to have high carbon dioxide emissions. So if we're succeeding in one area, sometimes that can end up having a balance out effect, which is why trying to mitigate nitrous oxide emissions while also keeping carbon dioxide emissions low instead of causing that inverse reaction is really important as well. So doing things like using nitrification inhibitors to help stop that one process but not increase the other one, or inoculating the soil with more microorganisms that could potentially reduce that nitrous oxide as well.
Great. Melissa, did you want to add anything there? No. Okay. Okay, thank you. Moving on to the next question. If each soil is unique in its composition and the subsequent effects of treatments for on-farm management, would each vineyard or orchard need to, I think this meant, it says rest, but I think it meant test their own soil to determine how to reduce emission? And I think that has been sort of an overriding finding from our work, that each soil is unique, and it is important to look at, well, to test, and perhaps when there are more methods, easier methods to use, to look at some of those populations that can contribute to nitrous oxide. Catherine and Melissa, I'm sure you can add more there.
Yeah, I think it's important to test your soil and know what you're coming in with. I think a really good example is that all the soils we worked with fall within the texture class loam, but they were still different enough to exhibit different reactions to the treatments, especially with urea. So we see that even though they're the same texture class, they still differ because they are different in those percentages of sand, silt, and clay, which is then going to affect water dynamics and things like that.
Yeah, I agree. From everything I have learned about soil is that it's not even just the composition of the inorganic particles, it's the organic ones as well. It's the fertilizers that you add. It's everything that makes up that soil creates its history. It's like an individual person. Everyone is different because of their experiences, but also just because down to mix, they're all different. So it creates just a huge array of different characteristics that we need to try and encompass for people to either get their soils tested to find out what might be optimal treatments for them, or we need to try and find enough data about enough of these characteristics that we can hopefully build some sort of schematic for people to determine what treatments to use.
Yeah, good, all good points. I'm going to move on to the next question now. Were there any crop types which increased denitrification genes, such as cherries over apples, etc.? I'm not sure if we can answer that.
We did
all of my microcosms were on apple, so I can't comment on that. But based on the research, yeah, because it's what crop type you have does change the soil microbial community. So you can expect to see differences between your crops.
Yeah, detritus from the crops themselves can leach certain sort of phenolics when they have fallen to the ground. The root exudates are different. They vary in their sugar contents. So all of those different things are going to contribute to that. Additionally, the fruits themselves can contribute to it. And the pits in some different stone fruits have been shown to be toxic to some microbial communities. So it can definitely vary from crop type to crop type.
Yeah, I don't think we're in a position right now to say whether cherries or apples are affecting that. But good question. That would be a good subject for further study. Next question, how these studies can help us farmers to change our methods and amount of fertilizers used for optimum production. I would say we're, as both Melissa and Catherine mentioned, we're still in the process of finishing off these studies. We have, it's quite time consuming to analyze for all of these genes, and COVID this last year hasn't helped. It's limited our access to equipment and our ability to work in the lab. But I hope by the time we've finished in the next few months, all of our assays, that we may be able to produce some sort of fact sheet and some at least preliminary recommendations for growers. And I think the amount of fertilizers used, you shouldn't be using more than is needed. And so again, that goes back to testing your soil ahead of time. There are some fertilizers release the nitrogen more slowly than others, and those may be a better option. I'm sure the others also have some suggestions there.
I think, oh, you go, Catherine.
No, go ahead.
I was just going to say, I think it's important for us to take into consideration the limitation that we have by using a microcosm. We can't directly relate that to what is happening in the field. So my hope is that what we've seen in our microcosms can initiate others to go into the field and collect more of this data and see what's happening in the field when they're applying that treatment to the system itself.
Yeah, very good point. And we did try to do some field work, and Catherine is still analyzing some of that, but there's a lot more that needs to be done. Obviously, what we've found with the microcosms is that there's actually quite major differences between soils that are still the same, in the same classification. And so there's just a lot of factors that need to be looked at. And then there's the temporal factor and the environmental factors. How much rain are you getting in a particular cropping year? All those kinds of things that that can have an effect. Catherine, did you want to add?
I was actually just going to talk about something that we didn't necessarily look at, but that I have seen a lot of previous studies having promise with. And I believe many orchards in the Okanagan already do this, but using fertigation does seem to be very effective with calcium nitrate, because generally fertilizers that are not applied in this method have an uptake efficiency. It varies, but between 50 and 70. Again, it varies depending on the soil types. That's a generalization, but the uptake efficiency of fertigation is much higher because it's closer to the root and it applies the fertilizer over a longer period of time. So it's not exposing all of the soil to all of the fertilizer at once. So I think that already is helping to reduce emissions in multiple orchards where it's implemented.
Thank you. I apologize. That's my phone ringing in the background. I'm just not going to answer it. The next question, I think Catherine can answer quite easily. Please say what DMPP is again.
It's 3,4-dimethyl pyrazole phosphate. It's a pyrazole derivative, which is actually usually used for antibiotic treatment. And it specifically sequesters the copper ions in the soil, which are used by ammonia monooxygenase to complete that first step of nitrification. So when you sequester that copper away, they just can't use their enzyme efficiently.
Great, thank you. Okay, and I think I'm seeing one last question here. Oh no, two more. From Duncan Barnett again. Plants also change the soil. So are there plants that can be grown in the soil under the trees that would reduce nitrous oxide emissions? And I think there is actually quite a bit of work that's beginning to be done on this, looking at different cover crops in the row. The use of, as we've talked about here, the use of mulches or composts directly under the trees can be helpful. Catherine, have you done any reading on that?
A little bit, though the results are kind of varied. There have been instances where people have used things like lentils, which are high nitrogen uptaking plants. They have nitrogen fixing symbiotic communities. And this can help different growers because once these plants have grown, they help to actually hold on to any nitrogen that might otherwise be lost by irrigation drain off into groundwater. But the complication with it is that once you mix it into the soil, it could increase nitrous oxide emissions while it's decomposing because it is very high in nitrogen. But additionally, it could also sort of hog the nitrogen from the plant that you're planting it near. So it depends. Some people have found it decreased nitrous oxide emissions and that the neighboring crop type was fine. Others have found that that was not the case.
Okay, yeah, it's once again hard to give a very definitive answer because there's so many variables that can affect things. One last question for each of us. Can each panelist tell us from their perspective what the most important finding of this work is? So I'll maybe start with Tanya.
So my part of this work was basically laying the groundwork and develop the method so that Catherine and Melissa have some tools in their hands to actually do those scientific studies. And
that was a very important step in the study. And in fact, I'll mention that the Canadian Journal of Microbiology has just chosen that paper that has just been published as a feature of the last year of their publication. So I think that gives you some indication that these methods are critical for understanding how these microbial populations are varying under different conditions, and then subsequently what their contributions can be to nitrous oxide emissions. Melissa.
I feel like a broken record, but I'm just going to go again with the fact that all of these soils react slightly different to the treatments. So I think the takeaway is that your vineyard or your orchard may react differently to your treatment compared to somebody like Catherine said, a kilometer away. And you know your orchard best. you test the soils, you know the history of it. And so you need to take all those things into account, how you want to go forward with treating your own land.
Okay, thank you. And Catherine?
Yeah, I have to agree with Melissa. That is definitely what we found. I also think that this has kind of illuminated the need for some sort of, institutionalized program that can allow people to test things that maybe they don't know are there yet. So maybe they want the abundance of different microbial communities tested, and just make something more centralized so that it's easier for everyone to access and they don't have to kind of try and figure out who to contact to get this done. But additionally, I think it was kind of interesting that in the Summerland soil, we did see a reduced relative abundance of the archaeal MOA, which is very indicative of other studies that have been seen. So archaea may be playing a larger role than originally thought in the emissions from these soils.
Okay. And I'll maybe just finish with, I think we've added some very new information with respect to nitrous oxide emissions in vineyards and orchards. I think it's clear to all who are listening that we still have a long way to go. We've mentioned the need for more field studies. These measurements for genes are actually quite expensive. It would be nice if we could come up with something that was cheaper so that it could be done more routinely. But that may come with time. And I think it's been just fascinating to see how nitrous oxide emissions can vary, and that we need to keep that in mind if we're wanting to be more sustainable in the production of fruits and grapes. So with that, I think we will finish up. If you have any questions that you would like to put forward, just you can send them to each of us. This slide shows our emails, and we really appreciate your attendance today and your engagement in the workshop. And I'd like to extend thanks to our presenters, Dr. Vogel, Melissa Larrabee, and Catherine Jansen, and also Jessica Lattis and Mairead Norton, who've been working behind the scenes to keep this running smoothly. And I also invite you to join us tomorrow on February 18th from 11 a.m. to 1 p.m. for our final installment on life cycle modeling of agricultural greenhouse gas mitigation strategies, bark mulch and irrigation water. And that's going to be looking at the whole system from the transportation of materials and their alternate uses. And so I think you'll find that very interesting. And finally, feel free to visit the BC Food Web at bcfoodweb.ca to learn more. And thank you all very much for attending, and have a great day. Bye-bye.